Okay, part two, let's go. So here's the little outline for the chapter. These are all the little sections of the chapter, and that's great. So let's go forward here and talk about commensals and pathogens. So these are both things that live inside of our body, and I guess pathogens are things that we make an immune response to. And usually there's a reason. The pathogenic things will quite often cause damage or suck up a little bit too much of our resources. Whereas at least some of the commensals end up doing something useful for us. Some of those bacteria will, for example, secrete a factor that helps us clot blood. But for the most part, we want at least the bloodstream to be absolutely sterile. There usually should not be any kind of bacteria in there. I know there are some people who have said that certain vaccines, like a BCG vaccine, might lead to some amount of effectively circulating bacteria, which might be helpful. But I think that's even a little on the risky side. So yeah, your immune system is good at knocking out all kinds of stuff, from little tiny viruses to great big parasites. Look at that nematode worm over there, and everything in between. Okay. And your immune system actually has a couple of different components, and you can think of them as organized in layers of defense. So at the basic layer, you've got things like your skin and your epithelium that are there to mark what is the inside of your body and the outside of your body, and keep the inside sterile and let, whatever stuff happen on the outside that needs to happen. That's fine. Just a barrier. Once you are on the inside, so there are going to be things like complements. So C3 is one of the complement proteins. And then there are things called defensins. It's a little protein that can actually go out and attack bacteria. Quite interesting. And Reg3 gamma is how you say this thing. This is another one of those antimicrobial proteins. And some of these are going to be made inside the gut. So we'll actually secrete these around the area where new gut cells come from to try and keep that area a little bit clean and free. And some of these are going to be secreted in, for example, in the bloodstream in response to infection. So we have different cells in the body that are able to make these antimicrobial proteins. And they're usually going to be specialized cells that actually produce these. Then your next layer is going to be your innate immune system, which is going to be cells like, so the granulocytes are your basophils, eosinophils, neutrophils, and, I left off one, mast cells. There we go. And natural killer cells and macrophages. So some of these come from the myeloid progenitor cells, and some of these come from the lymphoid progenitor cells. But these are all cells that don't change their DNA as they age. So these are cells that are born with the same DNA that they usually die with, and that's fine. But these are also cells that cannot adapt. They do not have necessarily a memory, although there are cases where you get some macrophages that have slightly different roles, but it's not really memory in the way that the adaptive immune system does it. Anyway, all three of these things are aimed at, the top three layers are aimed at buying time. They're aimed at buying time, and they're aimed at weeding out anything that we can weed out. So stopping any bad things before they happen. That's the idea. But if bad things do happen, then you're probably going to need to go to step 4, which is right down here, which is adaptive immunity. And adaptive immunity is your B cells and your T cells. Cells that will actually change their genetic makeup in response to whatever it is that has affected you or infected you. And these cells can then potentially remember winning combinations, whatever they've changed their DNA to. They can put aside some versions of that are called memory cells. And these things can live for months to even years, and can be reactivated so that you can preserve a winning combination and remake it again at a later date really quickly, without having to go through the random process that led to making it in the first place. Okay, so another way to think of it is in terms of the way that it's going to work. So you're going to start out with inflammatory inducers. So inflammation is this thing, and if you're involved in healthcare in some way, you know the signs: the pain, the redness, the heat, the loss of function, and I can't remember the redness. Did we already do that one? I don't know. Yeah, but anyway, those are the 5 external things that you can see. What's going on internally is that you're going to have compounds like histamine are going to be released. Proteins like interferons are going to be made. And these are going to be messages from one group of cells to another group of cells that say that basically there is something that is wrong here. So there are a lot of ways to induce inflammation. And they're actual components of bacteria, these things called lipopolysaccharides, that will induce an inflammatory response all by themselves without, any extra work on your part. And then if that fails, or part of the goal of that is to bring in some other cells and to kind of highlight that there is a general, that something is wrong. Something is out of regulation, out of whack, something has invaded, just something is bad. And so then you're going to call in some of these sensor cells. And the sensor cell's job is to grab and eat, physically digest whatever it is that's at the site. And then they have to go and take whatever it is that they have digested, and they bring it to the places where the white blood cells are going to get together. So these are going to be lymph nodes or the spleen in a human, particularly. And in these places, they are going to effectively burp up little bits of whatever protein or thing they've just swallowed. And they're going to show it to all the T cells and all the B cells that are gathered around and say, hey, anybody ever seen anything like this? Can you make something that maybe, will bind onto this? And so those cells will frantically recombine their DNA, try to make something that actually will stick to it. And then they can get a little sort of well done signal if they make the right thing, if it actually binds. And then from there, they can divide, they can make more copies of themselves, and they can go out and try and refine that, find different ways to bind the same thing. And then these things get turned loose, and they go and seek out that thing, whatever it is that the macrophage or neutrophil or dendritic cell has brought them. They will then seek it out wherever it is in the body and basically grab it and kill whatever's attached to it. So that's nice. And so these cells, macrophages, neutrophils, dendritic cells, for example, can produce little things called cytokines. So these are usually going to be little proteins that act as messages from one cell to another and can be used to call other cells into the site where something needs to be done. So when one of these cells gets a hit and finds something out of order, it can send off a signal that calls in other cells to say, hey, come and take a look at this. And then let's go report together. And at the same time, we can cause cytotoxicity, find ways to destroy any cells that are malfunctioning or infected. And then finally, the last step is to actually go in and target those tissues. So it's going to be mostly the adaptive immune system, but also some of these innate immune system cells are going to go in and actually make proteins that kill infected cells or that kill any bacteria that are in the area. But the immune system is kind of a brute force sort of creature, if you want to think of it that way. It will generally cause more destruction than is strictly needed to cure a problem. And then repairing this destruction often involves scar tissue that can last a while or can last forever, and temporary loss of function in whatever tissue they happen to have destroyed. So there is a thing called the blood-brain barrier, which is to say that the blood vessels that go through the brain are specially impermeable. So they try not to let these goofballs through, like macrophages, neutrophils, and dendritic cells. Instead, there's a separate kind of parallel immune system that operates in the brain. And instead of macrophages and neutrophils, you're going to have these astrocytes. And they can do a lot of what macrophages would do. And then if astrocytes need a little bit of help, they can call for other cells from the immune system to invade. But generally, the brain's going to be closed off because if you get these dumb macrophages and neutrophils all fired up in there, they can cause widespread destruction. And if it's a lot of neurons and you happen to need those neurons to, do stuff and think, then that would be a fairly bad effect. Okay, how long do these things take? Well, the innate immune response starts up. The best thing about it is that it's quick. It's a little hard to regulate. It's the thing that goes off and causes a lot of problems. Yeah, it's gonna start up within minutes, and it's gonna last hopefully just for days. When the adaptive immune response kicks in, it tries to turn off what's left of the innate immune response. That's one of the things that happen. So adaptive immune response cells, like these B and T cells, will send out signals to try and regulate and basically tell the macrophages and neutrophils to stand down. We're here and we've got this. But it bought time. That's the important thing. It buys time within those first couple of days. Within the first few hours, you will have interactions between antigen-presenting cells, like a dendritic cell or a macrophage, and the first T cells. And the T cells are going to be working really hard to try to make a combination that matches that new antigen. Same thing for B cells. Within hours, you're going to start trying. But it's probably going to be a couple of days before you get decent amounts of T cells or B cells, things that make antibodies, actually produced. And so the general rule is, with an infection, you're probably not going to see an antibody response until at least a week after the infection has started. But because the process of making B and T cells is random, they have to randomly change in ways that the cell doesn't know if it's going to make the right change, and there's no one to really reward it very much along the way. Although there are little bits of reward, there's a little bit of feedback there. But so these things are going to take days. Something like 7 to 10 days is pretty normal for you to get the first sort of vestiges of a proper adaptive immune response on the case and revved up. Then the nice thing is they can last weeks or even months. Once the immune response is there, it's going to go to town. It's going to get rid of the problem, whatever it is. And then afterward, some of these cells that you made that happen to do a really good job, these T and B cells, are going to be basically put into storage. They're going to turn into memory cells. We're going to activate a couple of proteins on them. One of those is going to be CD45, and that's going to be a marker that says that this is a memory cell. And it's going to, whereas a normal cell from the immune system would die within a day or maybe a couple of days, these guys are going to live for years at a time, potentially. And the immune response can be something approaching lifelong in some cases. But with this lifelong immune response, you are relying on a very small handful of cells. A year after an infection, you may only have 5 or 10 individual white blood cells that still remember that infection. And if you don't see that infection again, eventually those cells kind of wander away or get lost or will die of old age. And so you can go down and down and down, and you may get to a point where you only have 1 or 2 white blood cells that still remember, that cold you had as a little baby. And you'll get to a certain point where potentially those cells, the last cell has died off, and there's nobody that remembers it anymore. And in that case, if you get infected again, it'll be just like you had never had this in the first place. And you'll have to go through the entire process of making an immune response all over again. So the immune system still works. It just doesn't last forever. Yeah, doesn't even last an entire lifetime. So all right, let's meet these little guys. So these are some of the cells. You can see their little picture there. You can see a little bit of what it is that they do to try and keep you safe and healthy. And the drawing over here, this is how they're going to draw that kind of cell every time it appears in the text. So it's a very kind of visual book, and so if you got these down and you can kind of identify them on sight, this will help you understand what's going on in some of the figures. All right, so let's talk sensor cells. Now they have a lot of ways of actually sensing. So for example, macrophages are going to have receptors, which are proteins. And these proteins can bind a lot of different things. So glucan or mannose receptors are going to be looking for kinds of sugar that you would find on certain bacteria but would generally not find on one of our own cells. And so if they get a hit on that, that tells them, hey, something's wrong, we need to mount a response. It's time to go eat something and then show the rest of the immune system what we found. Toll-like receptors, that's what these TLRs, and the toll is TOLL, just like when you go on the highway. So this is toll-like receptor 1, 2, 4, and there's a whole bunch of these intracellular. One of the intracellular kinds is called NOD, but it's just another version of essentially the same protein. And what these do and what scavenger receptors do is they each respond to a different thing. So some of these things will go off when there's a bit of DNA floating past the cell. Because, of course, DNA is supposed to be inside cells, and if you have a bit of loose DNA, that means something bad has happened. It's like finding a big blood trail, leading out of your bedroom in the morning. You say, wow, yeah, I don't know what happened here, but it's something definitely very bad. And so these are the kinds of general signals that those parts of the immune system get. And when a white blood cell gets activated, like a macrophage up here gets activated, or here's our little neutrophil down here, and there's another macrophage, although I guess if it's still in the bloodstream, it's probably a monocyte. And here we've got, I think, a T cell, maybe, is the blue one. And we've got a red blood cell, that's the little pink circle there. And so these guys are going to first send out signals. So cytokines and chemokines are two different kinds of protein, and we give them different names for old historical reasons, but they're really similar. And they're going to cause other cells that are rolling along through the bloodstream. So these cells are circulating around the body every couple of minutes. Cells move really fast, and so there's always some kind of cell on patrol. And so if you send out a message, one of these cytokines or chemokines, it's going to percolate through and get up to the edge of the vein, and it's going to leak out into the vein or artery right here. And any of these white blood cells that senses those cytokines or chemokines will have the ability to potentially respond. And so what's going to happen is the cells that line the bloodstream are actually going to contract a little bit, and this allows fluid to go into the area. This makes it easier for cells to come from the bloodstream into the tissue. So this sort of pinkish area up here is going to be whatever tissue we're talking about. And so even monocytes like this will come in, and then they will get activated and turn into macrophages. And so you'll have, it's a way of calling for help and essentially calling in all the different parts of the immune system that can see what's going on and try to do something about it. And this influx of fluid and this calling in of different cells is what leads to the swelling, the heat, the redness, the pain, the loss of function that we associate with inflammation. So this is kind of where it comes from and kind of why it's happening. It generally helps the body to fight by making it easier for cells from your immune system to get where they need to be going. Okay, next little goofball on our little hit parade is going to be a natural killer cell. There are natural killer cells, and there are things called natural killer T cells, or NKT cells, that are somewhere between a natural killer and a T cell in terms of what they do and what kinds of gene expression they have. And so these cells, like macrophages and neutrophils and all the granulocytes, have these little tiny, see the little pink dots? Little granules that they can release. And these granules will contain different cytokines and chemokines, and also some proteins that will actually assemble and punch holes into cells. So they're actual little machines that can kill cells. And these are all the spare parts, and the spare parts just self-assemble once they are spit out by the natural killer cell. So those are kind of neat. All right, and so that's the end of that section. Then we'll come back here for the principles of adaptive immunity. And remember, this chapter is just an overview, but I want to get it, kind of in front of you as many ways as possible, the basics of what are going on. Once you can think like the immune system, it becomes a lot easier to understand what's going on in any situation, I think. So that's all for this section, and I'll be back with the next one.